All right, this has been a long chapter, but this is the last part, thank goodness. Cellular organelles, part 3. The cell itself, cells can have all kinds of different shapes. So we've talked about this before. Sometimes cells, it's beneficial for them to be highly folded. And the way they can have these shapes or even change their shapes is because they have a skeleton. And this is called the cytoskeleton. And there are 3 major components of the cytoskeleton. Here's an illustration from your textbook. You can see a cell membrane here, a cell membrane down here, and then you can see this tubular in green structure. That's a microtubule. This branching orange structure, this is the intermediate filament, and the actin filaments shown in red. Of course, the microtubule is going to be the big ones, actin is going to be the small ones, and so intermediate filaments get their name because they're intermediate in size. All right, so let's look briefly at each of these. The actin filaments are actually made up of these 2 long strands of proteins here. They're very small, only 7 nanometers in diameter. The 2 chains are twisted together. These actin filaments are going to be responsible for allowing the cell to crawl around, or in cell division, allowing the cell to pinch itself in half to make 2 cells. And these guys are going to be a major component in muscle contraction. But we'll talk more about that later when we study muscles. These guys are constantly being made and broken down, so polymerized and depolymerized. And so they have positive and negative ends, which designate the direction of growth. The positive end of an actin filament is toward the cell membrane, and the negative end of the actin filament is actually toward the nucleus. We'll talk more about that in a minute. We talk about how things move. All right, the largest component are the microtubules. These guys are 25 nanometers in diameter, and these are essentially hollow tubes. And you can see that they're made out of 2 different types of proteins, alpha tubulin and beta tubulin. I love that. Tubulin makes a tube. You know, if you look at the illustration here, you can see that we've got these dark spheres, and then next to that we've got these light spheres. So there's like an alpha tubulin and a beta tubulin, and the way they're laid down, they make this nice coiling tube. And then the middle size, one of the intermediate filaments, these guys are about 8 to 10 nanometers. And these guys are the most durable part of the cytoskeleton. Supposedly once they're formed, they usually don't break down. We're not going to talk much more about those guys, but the other 2 guys will show up again. Some structures that are composed of microtubules, we have one that's called a centriole. The centrioles are going to be found, we'll mainly talk about them being in animal cells and protists, but not found in plants or fungi, though we won't talk about fungi much. And you can see that a centriole is actually made up of triplets of these microtubules. So you can see we're going to have 9 triplets, 1, 2, 3, 4, 5, 6, 7, 8, 9 microtubule triplets. So the area around a centriole is called the centrosome, sort of the body, and then specifically an area that helps organize or put the microtubules together is called the microtubular organizing region. And again, we'll talk a lot more about that when we do the chapters on mitosis and meiosis. Now, this is kind of cool part here. It turns out that the vesicles that we've talked about, either the Golgi vesicles or the transport vesicles or the secretory vesicles, actually aren't just moving randomly. They're actually being hauled around by motor proteins. And these guys actually follow little tracks, and the tracks are, guess what, microtubules. And as you can see in the illustration here. And so this process requires a vesicle to be moved around. It requires some sort of a motor protein, which in this case, in the illustration here, is called dynein. And it requires a connector molecule to actually hook the dynein to the vesicle, and then, of course, a pathway of some sort. So the direction is determined by the orientation of the microtubule, which always has its positive end toward the cell membrane, and also by the type of motor protein. So we have dynein, which will move vesicles away from the cell membrane, toward the negative end of the microtubule, and we also have kinesin, which moves them toward the cell membrane, which is the positive end of the microtubule. And again, we have a really nice illustration that we'll show you as soon as in class this week from Harvard University that really shows this in action. It's just freaky and cool. A couple of organelles that are made of these microtubules are the cilia and flagella, and these are organelles of cellular movement. And in general, the flagella are long and few, and the cilia are short and numerous. Now, even though this looks like a lot, it's not. Look over here. Look at this hairy guy over here, this little paramecium over here. The cilia or flagella are essentially the same except for their length and number. They have the same 9+2 microtubular structure, and that is that, if you'll see here, they have 9 pairs of microtubules. So 1, 2, 3, 4, 5, 6, 7, 8, 9, surrounding two single microtubules in the center. And then between these pairs of microtubules, we find, again, those little dynein motor proteins, and they can walk along each other and cause the flagella to bend either one way or the other. We'll talk more about that in more detail that in class. These guys actually, inside the cell, we have a structure called a basal body, which you can see is composed of triplet microtubules. This one have the two central ones here. And again, this is what your illustration from the textbook's trying to show you. This is a basal body. And it turns out that that basal bodies actually kind of, in a way, anchor the cilia or flagella to the cell. So even though there's triplets here, only two of them persist into the cilium or flagellum, and then these two central microtubules show up outside of the basal body. So again, a basal body is a structure composed of 9 triplet microtubules, and those two of them are continuous with the cilium or flagellum. One thing that's interesting about the cilium and flagellum, even though it looks like it hangs out of the cell, this looks like something that's sticking out of the cell, in reality it's not. And if you look at this picture right here, you can see that the plasma membrane here actually surrounds the cilium and flagellum, and there's even cytoplasm up in here. So and obviously, if you're going to have motor proteins that are going to be walking around, you're going to need some form of energy, ATP. So these guys are actually still within the cell. So another little part we're just going to throw in, and this is plant cell walls. As you well know, these guys are composed mainly of cellulose. They're going to provide protection and support for the plant cell. And what I want to show you here is that they actually do have several parts. There is in this illustration here, you can see the cell membrane is this dark line right here. And what's secreted first, and again, that's given off for a useful purpose, is the primary cell wall. And it's fairly small in comparison to the secondary cell wall, which is given off second. And that's how the secondary cell wall ends up inside the primary cell wall. So, and then the plant cells themselves are cemented together with this area called the middle lamella, which would have to be secreted even before the primary cell wall. So primary cell wall is the first wall secreted. There's a thicker secondary cell wall that's secreted after the cell reaches full size. And then the middle lamella is composed of pectin, and that cements plant cells together. When I was a little kid, my grandmother used to make jelly, and a thickening agent in jelly is called pectin. It's the same stuff here. And there's a little note on, you already know this, but bacterial cell walls are composed of peptidoglycan, and fungal cell walls are composed of chitin. Well, that's it for now. A pretty short webcast. But if you have any questions, be sure and bring them to class.